Okay. Thank you very much for having me here, and it's a great honor to have a chance to present story of Telasabek, particularly in the event that organization that all the science has originated from. Okay, so just a brief introduction of Kiyeon. Kiyeon is a spin-out from the Institute Pasteur Korea, and our business model is called like a labless research house, meaning that we are not carrying a wet lab, but we are doing all these research and development through our network of academic institution, research institution, and the industry partners. And we are basically trying to address the unmet medical needs around the world, including the tuberculosis and the mycobacterium infection in this case. Currently, we are mostly focused on the oncology area, but this antibiotics, Telasabek is our flagship project that actually leads us to all the harnessing all the drug discovery development efforts. Currently now being developed by the TB Alliance based in New York to bringing the product into clinic fairly soon. So let me just briefly go about the Telasabek story. I think I am one of the few people actually remember the beginning and then tell the story about how it started. When the IPK was founded, there was a state-of-the-art technology present. Still, I think it was a high content screening facility in the BSL-3. This is the only high content screening actually set up in the BSL-3. So we were the IPK was the only institution can carry out this high throughput screening for these BSL-3 grade infectious diseases. So there was a assay developed through the scientists in collaboration with INSERM that the Priscille Brodin and the Thierry Christophe team actually were able to develop an assay that typical antibacterial growth inhibition assay, as well as that you are able to grow this TB infected in the macrophage. You monitor this through the imaging, and then you can identify specifically the macrophage that has been infected by the Mycobacterium tuberculosis. And upon the treatment of the compounds, you can actually measure the dying of this bacteria within macrophage with the right cause through the imaging process. And the assay was validated from the, existing, all these old generation antibiotics, showing that, this work is actually the assay works fairly well. And then there was a phenomic screen. It was titled as a phenomic screen. And this screening was conducted from the 2008 to 2009, basically screening about 250,000 compounds in collection to screen for to find out a novel compound actually that blocks this the mycobacterium growth in macrophage, also as well as in the outside culture. So there are several lead compounds actually being discovered through this. The long story short, this particular compound called IPA01 was selected as a lead compound. This wasn't the most active compounds, but this had the right property for becoming a drug, and then that has a fairly good, activity at this time was almost 2 micromolar IC50 in these assays, which was a good enough starting point which can actually carry out, further development. And it's a root candidate. This is an integral effort, actually collaboration between the IPK and Kiyeon to try to come up with an actual drug-like molecule. From the IPA1, it took us about 6 months to bring it out, the activity into the IC50 of nanomolar range. And we also took on, like another 18 months to generate almost like a 500 to 600 different derivatives to find the final candidate. Obviously, that the drug discovery is basically trial and failure endeavor. So there was a Q201 and Q202, and the third candidate, Q23, actually made to the end. So it becomes, like our clinical candidate. And the license to Kiyeon actually took place in the February of 2010 in the middle of the lead discovery campaign, lead development campaign. The mechanism of action, interestingly, that when we discovered this series of compounds, there were two other institutions in the world that actually came up with the similar, biology, similar discovery of the similar series of compounds. One was the University of Notre Dame in US, and the other was the Novartis Tropical Institute in Singapore. And we were slightly earlier than in development compared to the other two institutions. So Novartis actually gave up on their own development and basically that they decided to collaborate with us. That was a great collaboration actually in order to find a target. As this screening in the beginning was the high content screening that we didn't really know the target. Compounds works greatly, but we had no idea how this target works. And then in collaboration, Novartis had a clone that are the resistant to this particular, like series of compounds. And then we were able to test our compounds, candidate compounds against those clones. And then we were able to identify the exact target, the mutation that happened in that resistant clone. And then that was in collaboration with Pablo Vifani at the Novartis Singapore at the time. And then we identified the cytochrome BC1 complex as a target for this particular mechanism. We are quite surprised by the finding that this basically the targeting the energy metabolism, particularly the electron transport chain, has been used for the developing a pesticide. But we never thought that this could be a mechanism of action for the actual drug that doesn't actually affect the human body. But in this case, this particular series of compound was affecting only the electron transport chain of a mycobacterium, not human or any other bacteria. So the clone, this discovery was quite impressive. And then this is because first in class worthy of discovery that leading to a whole new series of antibiotics. As I mentioned, that is cytochrome BC1 inhibitor is the third complex of the electron transport chain of a mycobacterium. By doing that, by inhibition of this energy metabolism, telasabeg is actually able to block ATP synthesis, meaning that a lot of different antibiotics affect only the proliferating bacteria, meaning that if a bacteria goes into dormant state, then it cannot work. But if you block the ATP synthesis, then you can actually block the dormant bacteria as it grows as well. So telasabeg was showing very good activity in not only in the aerobic phase or in the anaerobic phase, the status of the bacteria growth. And obviously that this is a completely new mechanism of action for the anti-TB world. So we tested against quite a number of different human isolates of tuberculosis, including the drug-resistant one, MDR, XDR, or something called the TDR is totally drug-resistant. It's resistant against all 15 drugs that being used in human TB infection. And then the compound is new mechanism of action, so it worked for all of them. And interestingly, that the compound does not work for any other bacteria. Later, we found out that the compound is working for some of the mycobacterium, but at the beginning that we didn't think that the compound is going to be very, very specific for the Mycobacterium tuberculosis. So, in vivo model taken place in mouse. Basically, the compound was very active in vitro, and it was also proven very active in vivo and in a very dose-responsible way. That you can see that from the 0.4 milligram per kilogram to 10 milligram per kilogram, you can see almost like a 3-log reduction, meaning that 99.9% of the bacteria got removed from lung within 4 weeks of treatment. And this compound not only having an antibacterial activity, one of the very interesting findings was that we were able to observe that the inflammation caused by this mycobacterial infection is actually being reduced by the compounds. So you can see a lot less tubercle. This is inflammatory body actually being reduced by the compounds. We understood the mechanism is a compound is inhibiting the 5-lipoxygenase in weekly. So it was actually having an antibacterial plus anti-inflammatory effect. And then all these findings were published in the Nature Medicine at the time. And the chronic mouse model, the tuberculosis is a particularly drug-resistant tuberculosis. You have to have a combination regimen, at least the three antibiotics combination in order to prevent that the resistance occurring easily from the patients. So you have to test that what kind of other antibiotics actually have a additive or synergistic effect with this telacibeg. And we can show that the telacibeg is quite synergistic with the newly discovered another energy metabolism blocking agent, bedaquiline, developed by Janssen. And all these results actually, supported the telacibeg moving into the clinical development. And then we conducted the clinical development in the phase 1 in the US and the phase 2 in South Africa. So phase 1 study was done in the US, and then it shows a very good dose response in summary. And then there was a minimum of the adverse events found, and then its compound was surprisingly very clean. So it's a summary. So one of the things that we cared very much about was the cardiac tox, because a lot of these antibiotics was developed earlier ages that has a very effect on the cardiac arrhythmia, so that it causes irregular heartbeats. Could be very dangerous for the patients. So in combination regimen, we did not want to amplify these already existing risk. So that we screened against this ECG risk very carefully, and actually it didn't happen in human. And the summary of the phase 2, this called is early bactericidal the activity study, meaning that the TB is quite complex disease to begin with. So you will need to have at least 2 or 3 different drug combination for just regular TB as well. So if TB patient, new drug for the TB is identified, then what happens is that you have to recruit patient prior to treatment of the standard regimen, meaning that the patient has to take a risk of not being treated for the standard regimen for a certain period of time. So 14-day treatment was set as the ethical period that, delaying of the start of the standard period. So you have to demonstrate the efficacy during this 14 days window that this monotherapy of certain antibiotics has to work in that window of a period, which is very tough to achieve. The study was designed in this way, like from the safety study, the compound was safe until up to the 320 milligram per day. So we decided to have a 3 cohort, 100 milligram, 200 milligram, 300 milligram, and compare that with the standard regimen of rifampicin, which is containing the rifampicin and the isoniazid combination. So result within this 14 day was quite remarkable, that you see that the Telasebek activity basically being measured by the bacterial growth from the sputum of the patients. If a patient had a sputum, then you basically culture this sputum in the media to see that how long it takes to actually make a colony. Longer it takes, meaning that there are less of the bacteria, so longer duration of this time to positivity means that the higher activity. So you can see that 100 milligram, 200 milligram, 300 milligram, you can see the increasing trend of the time to positivity. So which is a really great sign. It's a good drug, typically response to the drug dose level. So 120, 300, in this 14 days of the very short period of time, response was quite impressive. And that is even compared to the 4-drug combination of the Arifa4 is rifampicin, isoniazid, pyrazinamide, and ethambutol. 4-drug combination is comparable to the 1-drug effect. So the data was remarkable, and then this was the first drug actually shown the statistically significant difference as a monotherapy in this 14-day trial setting. And then this result was published in the New England Journal of Medicine. So TB is being developed as a new combination regimen. And the later research, I mean, the Telasebek is becoming unknown for the wider scientific society, and then there are research and development actually going on for the other scientific world. And to our surprise, that the Telasebek was actually active in the other mycobacteriums called the Mycobacterium ulcerans that cause a Burley ulcer. And this Mycobacterium ulcerans is actually causing a quite, severe skin condition. This disease is mostly, widespread in West Africa as well as some part of Australia. This is something called the flesh-eating bacteria. So once you get infected by the bacteria, then you basically skin get deteriorated and necrosis happens. And you have to amputate if the treatment is delayed. The standard treatment is that the 2 antibiotics combination for 6 to 8 months. The issue is that the people don't come to clinic very often when you're infected by the bacteria, particularly if they experience the treatment of antibiotics, because this bacteria, as Dr. Jordan mentioned, that this particular bacteria create basically the kind of a pain medicine. So when this bacteria is abundant in the tissue, then patients do not really feel the pain. But if you take an antibacteria, then they start to feel pain, and then they don't come back to clinic to take another dose of the antibacteria. So what you need is a really fast acting antibiotics against these type of bacteria in order to cure the patient. And as you can see that their findings shown that this Telasebek shows an ultra short treatment of the Burley ulcer in mouse. That was quite encouraging, that the scientists were able to demonstrate that only 1 week of treatment can clear the entire bacteria, and there is not coming back from mouse. So we actually went on to test this in the clinic while we are trying to actually doing that. And then we decided to, actually, in order to make this a reality, our biotech based in Korea, and then this is the kind of disease that in the West Africa, in the different parts of the world, we are physically cannot reach to there, actually do a clinical trial in the West Africa. And we had to partner with an organization which actually can do this. And we actually made the agreement with the TB Alliance. the license agreement to TB Alliance, to having TB Alliance actually to carry on the rest of the part from the late stage development to the commercialization of these Telasabec to actually being able to reach to the patients. So TB Alliance took on the further development of the Brule ulcer. Then because I mentioned that this disease is prevalent in West Africa, as well as in the southern part of Australia, particularly the area near the Melbourne area. So we actually went to Australia in collaboration with the hospital called Barwon Health to see that whether this actually Telasabec is working against Brule ulcer infection in human. So pilot clinical trial happened is against this patient in Australia called the Treat BU trial. We first recruited 40 patients. And then in Australia, there is a season for this particular disease, which is typically happening in the winter time of Australia. And up to day 34, 40, 84% of the patient has been healed. We are not talking about the removal of a bacteria infection. Bacteria infection is already gone. The actual cure is measured by the horrific wound actually being healed to a normal tissue, and 84% was healed at the time of the presentation at the WHO last spring, that we were able to show that the drug is doing great after the 1 month treatment is being cured. And then later follow up, then we can actually confirm that 100% of the patients actually got healed completely. So only like 1 month treatment of the compounds, actually we were able to heal 100% of the Brule ulcer patients in this setting. So it was a great success in terms of the clinical development. And now we are moving into the approval study, phase 3 study, to bring this, like a drug into the patient's hand, and then we are running it in Australia first to show that this is the case. And later on this year, that there is a new international organization has been formed. It's called the TEBLA. It's a Telasabec to control Brule ulcer and leprosy. The reason why the Telasabec works fairly well to the Brule ulcer is one of the reasons that there is two overlapping pathway in the electron transport chain, cytochrome BC1, and then going through the BC complex. There is a sidetrack of the going through the BCD complex. TB, tuberculosis, has both pathway. So if you block major pathway, then it retards the growth and then finally kill the bacteria. But this Brule ulcer, Mycobacterium ulceran, or the leprosy-causing bacteria, Mycobacterium leprae, do not have the alternative BD pathway. So if you block this BC pathway, then it completely blocks the energy metabolism. That's why that, it works fairly well to the Brule ulcer, and which is also the case for the leprosy. So there is an international organization has been formed, backed by the EU, to actually develop Telasabec to address these two particular disease condition, which is very prevalent in the less developed part of the world. It's called TEBLA. So all this, I think, is actually going in now, is currently moving on, and then we expect to have these compounds start being used commercially in patients from as early as 27 next year. Then we can actually finally, that the research project has started in 2008 in Institute Pasteur Korea, actually coming to a market almost like 20 years later to reach to the patient. This is the kind of lifespan that it takes to develop a drug, and particularly the drug that has a very long unmet medical needs for the patient population who cannot afford the drug. So this is the, very much of the international endeavor, collaborative endeavor. The project is, started from the Institute Pasteur Korea in collaboration with INSERM and Novartis. TB Alliance is taking care of the later development. Baron Health is now is running, a bully ulcer trial. Task and the University of Cape Town are the one who conducted the TB trial in South Africa. Such a international effort. There are many more institutions actually took part in this journey, and the Korea Drug Development Fund actually funded the program from the preclinical to phase 2 program. And thank you very much for your attention.